Hello, am I audible?
You're audible. Yeah.
Yeah. So I think we'll start the session. Very good evening and a warm welcome to the seventh session of Student Journal Club and Networking Program. This is Bipasha, your host for the session. The guest speaker for today's session is Mr. Homen Phukan, SRF Fellow from ICMR-Regional Medical Research Center, Port Blair in Andaman and Nicobar Islands, India. He has completed his PhD thesis from Pondicherry University. He specializes in medical microbiology, proteomics, biochemistry, and cell biology. Today, Mr. Phukan is going to enlighten in the topic exploring outer membrane proteome and subcellular fractionation in pathogenic leptospira through cutting-edge proteomics. A quick announcement. Any questions during this session can be left in the chat box and will be answered at the end of the session. Over to you, Mr. Phukan.
Okay. Thank you, Bipasha, for introducing me. And it is an honor for me to deliver my work in front of you people. And let me share my screen. So, is it visible, my screen?
Yeah, it is visible.
Okay. And very good evening to all of you. And I'd be discussing about the outer membrane proteome and how to extract this outer membrane protein with the help of subcellular fractionation with respect to pathogenic leptospira. And further, how we perform this outer membrane proteome analysis with the help of proteomics. As you can see, bacteria having subcellular compartments, cytoplasm, inner membranes, periplasm, and outer membrane. And periplasm is having a small layer of peptidoglycans. This gram-negative bacteria have a double membrane that functions as a both protective for providing nutrients availability. The outer membrane comprises completely integrated membrane proteins that help the cell with food intake, cell adhesion, signaling, and export. And many pathogenic bacteria use the outer membrane protein as a virulence factor, and they used it as a host defense mechanism. And they can be maintaining the bacterial cell integrity. They can attach to various substrate like extracellular matrix components. And they can import nutrients from the environment and from the host itself. And they can export bactericidal agents and toxic agents. And most probably, they are putative vaccinogens and diagnostic candidates. The outer membrane acts as a first line of response regulator to external stimuli. And you can imagine like a situation of border where the defense people can be termed as the outer membrane proteins. These outer membrane proteins are required because pathogenic bacteria depends primarily on the membrane proteins during infection. To identify these OMPs and characterize them, it's very important for understanding the bacterial pathogenicity, and with the help and further we can develop any therapeutic interventions. It can be a vaccine, diagnosis, early diagnostic candidates. As you can see, the membrane itself, it is a quite asymmetric bilayer where now phospholipids are present in the inner leaflet and lipopolysaccharides are present in the outer leaflet. And this lipopolysaccharide is essential for its the structural viability. And lipoproteins are present in between this outer membrane and inner membrane, that periplasmic space, and also it is present in the surface of the outer membrane. And it is important for biogenesis of the outer membrane and involved in various cellular functions. And this outer membrane act as some kind of permeability barrier for many molecules. So it can allow the nutrients and other important molecules to come in the cell. And while at the same time, it can be a permeability barrier for against some other molecules like antibiotics and toxins. These lipopolysaccharides contribute its permeability and resistance to the external agents. Because of these functional barriers, it is very difficult to extract the proteins that present inside the outer membrane, inside the outer membranes. Then many scientists are getting challenges by extracting the outer membrane protein. Since the outer membranes are a crucial, outer membrane proteins are very crucial for the structural stability of the outer membrane. And it is very much careful need to take while extracting the proteins, which has to maintain its the proteins structural integrity as well as the membrane. And problems comes while it is like how to differentiate this inner membrane and outer membrane, because of they are having same kind of hydrophobic properties. Sometimes this phospholipid bilayer makes a complications. And outer membrane proteins are mostly hydrophobic proteins, which have a behavior of during the extraction process, and they tend to be partitioned into the lipid phase of detergents. And sometimes some kind of hydrophilic molecules also create problems during extraction process. And during extraction, another kind of problems comes to the cells, and that osmotic challenges encountered during the extraction process, which affect the integrity of the outer membrane and the stability of the proteins. Challenges using different kind of detergents. So detergents are essential for solubilizing membrane proteins, but the choice of right detergents can be significantly impact the stability and functionality of the extracted protein. So commonly used detergent like SDS, Twins, Tritonex, and with the addition of some additives like urea. So what they exactly perform during membrane protein extractions, they disrupt the naturally associated lipid bilayers, which ultimately lost the protein subunits, and it's affect the integrity of the proteins, and which ultimately affect the downstream processing or downstream analysis of the extracted proteins. Even you optimize the detergents for membrane protein purification, then it is largely empirical, and often it's resulting in the failed in preparation, and it is a high cost. Alternatively, if you increase the concentration, it can readily solubilize the outer membrane proteins, but it can cause the ultrastructural derangements, and ultimately it will affect the specific activity of enzymes. Further, these proteins are very they want to be stay in the native lipid environment. So because of that, its delicate balance is required during the extraction process. So therefore, what kind of detergent we can use for our desired experiment to extract the outer membrane proteins? The detergents mimicking a lipid-like environments are promising for outer membrane protein extractions, which can influence the solubility as well as it can preserve the native conformation of the and stability. In that sense, the right detergent can be Tritonex 114. It's a kind of non-ionic detergent, and some non-ionic hybrid detergents. So I will be talking about this Tritonex 114 detergent, which has a ability to differentiate hydrophobic and hydrophilic phase partitions, and which can efficiently extract the integral membrane proteins, which are hydrophobic. So it can act as a kind of biological membranes with while we use some certain factors like temperature, and it can get separate by with the help of temperature like hydrophilic and hydrophobic portions. Hydrophobic, nothing but it is a detergent concentrated forms, and the hydrophilic portions are polar. So let me discuss about these biphasic separations. Being a surfactant, it is amphiphilic molecules and composed of hydrophobic and hydrophilic portions. And one of the most important essential biophysiochemical properties of this amphiphilic molecules has critical micellal concentration, which is nothing but the minimal surfactant concentration required for forming a fully functional micelle. This micelle helps in the solubilizing of the membrane embedded proteins. Another important factor is cloud points, which is nothing but the temperature above dilute aqueous solutions become turbid, and eventually it can form a two phases. So you can see the picture itself. So here you can see the during the temperature 0 to 20 degrees centigrade, you can see a monomer of the Tritonex 114 detergents. And so once we raise the temperature, it started forming a monolayer. Then once it cross the temperature, it will form a it will form a fully functional micelle. So if we use this particular detergent for extracting the membrane outer membrane proteins, what it will act, it will create a mycelial solutions, and it will mimic the biological membranes, and it will solubilize the first line of defense, which is outer membrane proteins, and it will solubilize with the help of, it will form a like detergent proteins mixture of micelles. At the same time, it can penetrate the inner membrane also, but because of this periplasmic space, it has very less access to penetrate the inner membranes, and it is unable to hydrolyze all inner membrane proteins. And because of this penetration, that cytoplasmic proteins may comes out, and it will get solubilized with the extraction buffer. And further, if we increase the temperature above the cloud points, so it will form a biphase, which is detergents and aqueous solutions. Aqueous is hydrophilic, and detergent is hydrophobic portions. And hydrophobic portions can be called most of the outer membrane proteins get solubilized in the hydrophobic portions. So because of that, this particular detergent is quite cost effective. With a mild concentration, we can use for extraction of the proteins, as well as we can get all subcellular compartments in distinctly. So how we use this entire approach for our study? So we perform a study in the Leptospira, that how the outer membrane proteome of Leptospira responds to host physiological conditions during interactions. And this work was recently published in the Zephyr. Let me introduce with leptospirosis. So leptospirosis is nothing but it is one of the reemerging zoonotic disease. It is spread through the infection by pathogenic Leptospira, and it is emerging due to the growing number of population residing in the urban slums, as well as increasing the frequency of climate change in the tropical and subtropical regions. So earlier, this disease was also termed as an occupational disease, because who were farmers going to the paddy fields, they used to get infected. But the situations now totally changed. So you can see the rodents are primary reservoir of this organism, and the bacteria comes with the passed through the urine from the reservoir host. It will come to the contact of water bodies as a kind of stagnant water, where you can see in the slum area and all, where the very poor sanitation and hygiene is there because of that. And what happens this during sudden flood and hurricanes, these hiding pockets will get splashed with this flood water, and it will come into the contact with the water bodies. And so this humans can be called as an accidental host, because if they are having cuts, wounds, and this is not healed properly, and if they swim also, so it can go to their body, and at a particular certain incubation period, it will establish the infection, and it will show the symptom. So the critical point is that these symptoms are very difficult to detect whether it is can be because of Leptospira or something else, because it mimics other disease like malaria, dengue, and other hemorrhagic fevers. The patients come to the clinic, and then physicians mostly misdiagnose these patients, and because of that, after a certain period of time, their or multi-organ get failures, and patients readily finally gets die. And Leptospira is being a spirochete. It is a spiral shaped, and it is having a 0.15 diameter. It's and 10 micrometer its length, and you can see a cross section. It is having outer membranes and inner membranes, and in between the peptidoglycan layer in the periplasmic space. And during invasive dissemination, this bacteria will come into contact with breast skin, as I already mentioned. Then it will pass through the epidermis. Then it will pass through the basal membranes, and it will try to degrade the adjacent endothelial cells extracellular matrix, and it will pass through that, and it will further disseminate to the other organs through the circulations. So during that, the leptospiral outer membrane proteins play a crucial role regulating the response to host environment. How? Because they might mostly get differentially expressed, and which enable the microbes to adapt and thrive in the host environment, and which can enhance its infection potential and resulting its disease. And Leptospira can be either pathogenic or saprophyte. The pathogenic, mostly it can cause disease in animal and humans, and saprophytic doesn't cause any disease. And there are more than 200 serological variants. And its incubation period, I already mentioned, like 5 to 14 days, and it can go to also 2 to 30 days. And estimates indicate that there are more than 5 lakh cases of leptospirosis per year worldwide. And our lab were continuously working from past 10 years. That the earlier work on subcellular proteomics analysis using Tritonex 114 fractionation showed a higher abundance of outer membrane proteins on the outer membrane, and most of the pathogenic proteins they could able to find in the inner compartment. And it hypothesized that this pathogenic proteins abundance may increase while these bacteria encounter in a pathogenic environment. So during interaction with host, Leptospira encounter 2 important host physiological parameters. One is temperature, and another is osmolarity. So you can see Leptospira mostly present in water bodies, and the temperature goes from 25 to 30 degrees centigrade, and osmolarity 60 to 66 milliosmolar. When it comes into the contact with a human body, it has to encounter the human body temperature, that is 36 to 37 degrees centigrade. Even it goes to 39 also in febrile phase of illness. And another important thing is osmolarity is 300 milliosmolar, which is 5 times more than its ambient environment. So during that situations, the proteins are the main important molecule for this tiny organism, which help the organism to survive in the host, and which will allow the organism to establish the infection. So therefore, osmolarity and temperature are considered important significant factors for Leptospira transition from free living to invasive phase. And the changes in the pathogen proteome may be key important player for their survival in the host. And many studies were going on how the Leptospira proteome responds to environmental variables, but still their pathophysiology unclear. In such case, it may be possible to pinpoint a virulence factor interacting with the host during infection. And in our lab, we mimicked the pathogenic conditions, and we exposed the cultured Leptospira, as you can see in the figure itself. We exposed them to a raising of temperatures, that is 37 degrees centigrade, and we osmolarity remain same. Then we exposed to a osmotic condition, that is 300 milliosmolar. Then temperature remained same as normal standard culture conditions. And we use both factors together to see the changes and all. Then we took the harvested the cells. We perform Tritonex 114 fractionations, subcellular fractionations, you can see. Then that is aqueous fractions, detergent fractions, and pellet fractions. And further, we proceed for trypsinization, and we label the peptides with tandem mass spec labeling, and further we subjected to LCMS analysis, and we acquired the data. This is methodology only. You should not get confused with this terminology, though. Let me explain. This PT, nothing but is a physiological temperature, which the temperature we raise to 37 degrees centigrade, as you can see here. Then the physiological osmolarity is termed as a PO, because I would be explaining later for upcoming slides. So because of that, okay, I'm explaining here. And 150 millimolar sodium chloride we used for mimicking the osmotic condition, that is equivalent to 300 milliosmolar. And both synergistic conditions we put together, and that is standard culture condition we kept as a control. And let me explain a little bit about this Tritonex 114 fractionation for the Leptospira. So we, after harvesting the cells, we used 1% Tritonex 114 buffer. We kept, we just simply gently mixed the cells with the extraction buffer, and we kept for overnight at 4 degrees centigrade. Further, we proceed with centrifugation. Then we took the supernatant, and that supernatant, then cell pellets we kept as separate. Then that supernatant, we again used a sufficient amount of another Tritonex 114, and we will make the volume concentration 2%. Then we kept in a 37 degrees centigrade for 1 hour. Then it will get distinctly separate the detergent phase and the aqueous phase. At the same time, we extract the inner membrane proteins by using pellet extraction buffer, which is nothing but we used urea, 8 molar urea, and we termed as a pellet fractions. And further, we proceed for mass spectrometry analysis and sample prep. We took the samples and in solution trypsin digestion and peptide samples. Then we performed the TMT mass spec labelings and chromatography, further process for LCMS analysis, and we took the data and submitted the database. And data analysis, and we perform a statistical analysis involving data filtrations, log to data transformation, data normalization by slope, imputations, and p-value calculations. And proteins having statistically significant differential expression, we took a significant value less than 0.02. Now findings. What we found from our study, we could able to find nearly 3,146 proteins of Leptospira, which is accounting 86% of the predicted proteome of Leptospira of their database in NCBI. There are nearly 2,000 proteins in the aqueous fractions, and 800 proteins in the detergent fractions, and 2,700 protein in the pellet fractions. And we could able to find the Leptospira proteins were differentially regulated with response to the two important physiological conditions. So we used a cutoff of fold chains is more than or equal to 1.2 as a up regulations, and 0.8 as a down regulation, and with significant p-value is 0.02. Then you can see in the here figure itself that in the total proteins and aqueous fractions, pellet fractions, and detergent fractions. So here you can see the in the detergent fractions that differentially expressed proteins showing significantly comparatively more significant to other fractions. And we perform qualitative and quantitative analysis of the differentially expressed proteins. Then while we accounting the total proteins for quantitative analysis for the differentially expressed proteins, and we could able to find 10% proteins were differentially expressed. And while we took subcellular compartments for the quantifications, we could able to find 14% proteins were differentially expressed. They are including both up and down regulations. And we could able to we try to figure out the number of how many number of proteins differentially expressed among the all fractions with respect to the treatments. And you can see here that that pellet fraction is showing highest number of differentially expressed proteins apart from the total proteins. Then but exceptional found in the when physiological temperatures are used. But you can see in the detergent fraction, there are the uniform numbers of differentially expressed proteins. Sorry for this. Yeah. You can see that in the detergent fractions, a uniform number of differentially expressed proteins, which including both up and down regulations. And further, we perform a comparative analysis with respect to this differential expression. We took up regulation and separately we analyzed, and the down regulation and separately we analyzed. And here you can see in the aqueous fractions, these proteins in different treatments, like in the physiological temperature, physiological osmolarity, and physiological temperature osmolarity, they have very variations. And similarly, you can see in the pellet fractions also. But if you look into this detergent fraction, we could able to find a uniform level of upregulation in all treatment groups, and similarly in the down regulation. So it's a kind of uniform level of differential expression. And further, we try to narrow down what are the proteins exclusively abundant and upregulated in the outer membranes. For that, we perform another comparative analysis by using only the upregulated proteins in each treatments, and we analyzed with respect to the fractions. So you can see here that 66 proteins exclusively upregulated in the detergent fractions in physiological temperature. Similarly, the 61 proteins exclusively upregulated in the physiological osmolarity, and the 60 proteins exclusively upregulated in the both synergistic conditions. Then we try to figure out this all proteins, whether they are commonly present in any other due to any other treatments, like physiological osmolarity, temperature, or synergistic conditions. Then we could able to find a total number of 88 proteins this exclusively upregulated in the all treatment groups. And from there, we could able to find that 14 proteins are common to all treatments. And these 14 proteins were considered for further analysis, as they are an important outcomes of 3 distinct in vitro physiological conditions of infection. And these proteins are likely leaders for strongly correlates their higher abundance on outer membrane during an infection. As you can see the table itself, these detergent fractions and aqueous fractions. So these proteins, suppose these proteins, it's significantly upregulated during these particular treatments, but at the same time, these proteins were significantly downregulated in the cytoplasm, and it can be found in for other proteins also. Further, we proceed for their pathogenicity analysis. We perform a bioinformatic pathogenicity analysis with the help of MPT, which is based on hidden Markov model and SVM algorithm, which can differentiate them the pathogenic and non-pathogenic cluster. So we could able to find among the differential expressed proteins, 24% are pathogenic proteins and 76% are non-pathogenic proteins. And further, we took 3 parameters for a comparative analysis. We took their pathogenicity, subcellular fraction, and differential expression. And you can see here that the upregulated proteins in the non-pathogenic cluster, we could able to find a higher percentage in the pellet fractions and the aqueous fraction, which is nothing but cytoplasmic proteins and inner membrane proteins. But if you look into this detergent fraction, the upregulated proteins could have, was found in the pathogenic cluster. It's a higher percentage compared to the other fractions. And another things, we have taken the 41 proteins for further analysis, which are common to all treatment groups. So from there, we could able to find 41% proteins were pathogenic and 59% were non-pathogenic. Even though this pathogenic percentage is less, their relative abundance, some of their relative abundance found to be higher in pathogenic cluster compared to the non-pathogenic cluster. Collectively, the results showed an elevation of leptospiral pathogenic proteins on the outer membrane in response to host physiological conditions. So as you can see from this heat map I have shown earlier also, that 41 from the 41 proteins, 17 proteins were pathogenic proteins, and which are likely to be a potential virulence factors. And this is the tables. And we use these potential virulence factors for a conservation analysis among the leptospiral species. So where we perform the conservation analysis bioinformatically, at the same time we perform immunoblot analysis with the, by using the specific proteins from the species of Leptospira. So this P1 nothing but is a pathogenic type 1 species of Leptospira, and which is highly pathogenic. And P2 is a P2 pathogenic species of Leptospira, which is you can say is a intermediate or comparatively less pathogenic. And S1 and S2 are saprophytic. So we could able to find most of these 17 potential virulence factors are highly conserved in the P1 pathogenic species, and this immunoblot also provide significant result. And we perform a validation for this differential expression of the proteins, exclusive proteins. That we took Lepel 41 and Lepel 46 as a model proteins for the analysis of immunoblot, and we could able to find a confidence results from the immunoblot. So you can see this Lepel 41 in control. It is having very less expressions, but once we expose to the all treatment groups, its expressions getting increased. You can see in the mostly, since we are targeting the outer membrane, so you can see the detergent fraction is getting, it is expressed high comparatively the control. And similarly for Lepel 46. So the validation indicating an upregulation of the selected proteins on the outer membrane during leptospiral infection. And from this selected potential virulence factor, this LIC2035 is one of the important protein, outer membrane proteins, which can interact with host extracellular matrix components. And its recombinant forms showed a potential serodiagnosis. And another important protein is Lepel 46. It is also surface exposed outer membrane proteins, and it interact with the host plasminogen and generate plasmin. And ultimately, it helps in the immunoevasion properties. And another important protein is Lepel 36. It is a thermoregulated protein, and it is relevant for host adaptability. And Lepel 41. It is one of the third abundant outer membrane proteins, and it is conserved in pathogenic Leptospira species, and it is readily solubilized in the hydrophobic detergent micelles, and it can bind with the host extracellular matrix components, preferably fibrous proteins. Another important protein is ERP-like lipoproteins. It is one of the potential virulence factor of Leptospira. Earlier, many studies they found these particular proteins, but they stopped working on it since this upregulation was not found in immunoblot. But now many studies came out with these proteins that it can bind with ECM components. It has immunological reactivity to infected sera, and it's a complement-mediated immunoevasion also. So with this finding, these proteins is likely to be a potential candidate for intervention of leptospirosis. We perform gene ontology and pathway analysis for these differentially expressed proteins, mostly metabolic process, molecular functions, and cell. And we could able to find many pathways with the help of KAG analysis. This KAG pathway analysis found that a smaller number of pathogenic proteins enriched for each pathway in each set of differentially expressed proteins. Since our interest on the upregulated proteins, and they may be directly or indirectly involved in the many pathways. So we could able to find many pathways related to these upregulated proteins. including protein export, cell motility, two-component systems, quorum sensing, metabolism, including carbohydrate, lipid, amino acid cofactors, and synthesis of secondary metabolites, including antibiotics itself. So it is ultimately suggesting that metabolic genes expression has been reprogrammed to adapt the host environmental changes. So with regarding this metabolism, you can see Leptospira breaks down long chain fatty acids instead of glucose or other carbohydrates in their host environment. So it is necessitates the expression of fatty acid metabolism proteins. So because of that, we could able to find that proteins were upregulated. Similarly, this bacteria may lower their oxygen requirement during host cell interactions by downregulating oxidative phosphorylation proteins, including cytochrome oxidase subunits, NADH dehydrogenase, and so on. Interestingly, the upregulation of acyl carrier proteins is likely responsible for fully activating the biosynthesis of several antibiotics. So this is a surprise for us because during these harsh conditions, this bacteria is expressing expressing the biosynthesis of antibiotics. But this surprise it did not last long because we could able to find the reason why it is happening. This antibiotics biosynthesis may be triggered by environmental oxidative stress or phosphate limitations. And in those cases, antibiotics are believed to be regulate the energetic metabolism of the producing bacteria. And we found many signal transduction, the proteins related to signal transduction, chemotaxis, export systems, cell motility, quorum sensing, and membrane transport, and more. And if you recollect the earlier table, that we could able to find lot of number of lipoproteins which were exclusively upregulated in the detergent fractions with respect to the treatments, and there are like 24% are lipoproteins and other are non-lipoproteins. Then it is giving an important clue why this set of proteins is getting expressed and coming to the outer membrane. So there could be certain kind of export systems in this organism. So Leptospira is having type 2 secretion systems, which is not found in any other spirochete, including Borrelia and Treponema. So it could be it could be utilized to secrete the surface lipoproteins, and it is in partially unfolded form. And we could able to find the differentially expressed proteins related to this type 1 and type 2 secretion system. And this and another component also we could able to find in sec translocate system also. So this analysis indicates changes in the physiological osmolarity and temperature, and hence the leptospiral protein export systems, including type 1 secretion system, type 2 secretion system, and sec translocase system. And collectively, the entire study signifies the importance of pathogenic proteins that need to be upsurged, and the non-pathogenic proteins need to be downregulated on the outer membrane during infection. See, if you see the conditions of this tiny organism, the being having the entire genome, and which can be expressed all proteins together, but this organism is not doing this kind of functions because this organism having a very limited resource to synthesize all components or proteins at a particular time. At the same time, it has very limited surface to make them allow to sit on the outer membranes. So because of that, it will allow only those particular proteins which are functionally important for the particular situations and which can sit on the outer membranes. So Leptospira also is more likely to utilize this strategy in its virulence mechanism. And our study could identify 17 pathogenic proteins I already mentioned, which are exclusively upregulated in the outer membrane and common to all treatment groups. And their high confidence in the conservation of orthologs also provide another additional evidence for their virulence. Overall, further research is needed for fully understand the role of those proteins in Leptospira pathogenesis and to explore their potential as a target for early diagnostics candidate or therapeutics. and this is references. And I would acknowledge my guide and supervisor, Dr. M.G. Madanan, scientist E, ICMR-RMRC Port Blair. So he helped me and in entire work for fulfill my this entire PhD work. And I would thanks to my colleagues and my junior. And I also thank ICMR for my fellowship. And I would like to thank Yanapaya University, particularly Dr. Kashyap Prasad for their mass spectrometry facility. And last, I would like to thank bioanalytics team for these innovative programs, Students Journal Club and network programs, and from the students and alumni of IIT Bombay. And thank you all. And I would love to take any questions you are having.
Thank you, Mr. Phukan.
Thank you.
I can see a question in the chat box. What software you used for KEGG and Go enrichment analysis?
It's a Panther analysis, Panther pathway we used for gene ontology analysis and KEGG. And we took help from a string database also.
Okay. Any other questions?
Hope I could deliver my talk properly for your understanding.
I couldn't get you.
No, hope I could deliver my talk for your for all understanding.
Yeah, it was very informative. Like it was in details.
Okay, thank you.
Yeah. I wanted to ask a question.
Yeah.
You said you analyzed the pathogenic and non-pathogenic proteins.
Correct.
So the non-pathogenic proteins were from the pathogenic strain or from non-pathogenic strains?
This Leptospira interrogens is a pathogenic species of Leptospira, and it is having both types of proteins. That means pathogenic protein also, non-pathogenic protein also. And things is that while it is encounter a pathogenic conditions, so this pathogenic proteins mostly important for its survival and making establishment of these infections. So both kind of proteins is present in this organism and this particular species.
Okay.
Okay.
Thank you.
Welcome.
Any more questions from anyone? I think that was all.
Okay.
Thank you so much, Mr. Phukan, for this informative session. Yeah, I think I am sure all of us we gained quite some knowledge from your session.
Thank you so much. Thank you all of you for attending this session, and I wish for this particular initiative of this program should go long.
I have one more question here in the chat box. How is LipL36 graph increasing as compared to LipL41? In LipL41, it is increasing continuously, but not in LipL36.
In terms of this figure or something else, can you repeat the questions?
Yes. How is LipL36 graph increasing as compared to LipL41? In LipL41, it is increasing continuously, but not in LipL36.
I think this is not LipL36. This is LipL46, and because I used that immunoblot analysis, so you can see the figure. I think, is it visible, my slide?
Yeah, the slides are visible.
Okay. Yeah, I think these slides you are asking. So LipL41, we took after immunoblot analysis, we performed normalization experiment. After then, we could found this it's a high expression with compared to the control. But in LipL46, we also found in the detergent fraction, you can see, but we performed this immunogenicity analysis for its density of the bands, then it will, after normalization process, it could able to give the proper results that this protein is getting expressed, that means comparatively higher than in the other physiological treatments. But it is mostly significant in the physiological temperature, as you can see. So this is the answer. If I correct.
Yeah, thank you. I guess the answer is understood.
Yeah.
I guess there are no more questions. Yeah. Thank you so much, Mr. Phukan.
Thank you. Thank you so much. Thank you, all of you.
You can leave the meeting now.
Thank you.